Hi, I'm Kevin Borland, and if you have been paying attention to the Facebook groups dedicated to DNA or following any of the genetic genealogy blogs online, you'll know that this week all the rave has been about a new tool by Genetic Affairs that does clustering. And that is, it takes a look at shared matches that it scrapes from Ancestry. And I think FTDNA it'll do also. And it'll organize them based on the shared matches of your matches and cluster them into a nice diagram. And you can use that diagram sometimes, depending on if there's endogamy in your particular situation. If not, or not too much, then you could use those clusters to uh, represent ancestors. And let me show you, let's take a look at the one I did using the genetic affairs tool, because it's really kind of neat. So it clustered my matches on ancestry.com like so. Um, and each of these rectangles, I was actually, by looking at what the cluster represented, who these matches were, I could take it down to a specific ancestor. And these are ancestors um, going back as far as my maybe 5 or 6 generations sometimes. Ole Peterson, he was my great grandfather on my Norwegian side. Charles Lindauer, he was also, I think, my third great grandfather on my Alsatian French side. Anna Kershaw. Um, but it's nice, and you get some structure within them too. And I think there's potential to go further back. Like, look at this one here, Peter Rice. He's my great grandfather, but there's sort of 3 mini clusters within that same cluster. And I'll bet you one is for Peter's father, one's for his mother, and one's for maybe one of his grandparents. Um, so, anyway, it's a really nice tool. Um, and they've done a great job because there is a similar tool on GedMatch. However, it's limited to the database of GedMatch, whereas the Ancestry database is a lot bigger. However, we're going to use the GedMatch tool because it's actually kind of more powerful, even though the database isn't quite the same size as Ancestry yet. Hopefully, it will be someday. But even now, there's a similar tool there, and let me show you how it works, because it not only works in a similar fashion, but it can be applied to DNA reconstruction, which is sort of my area of expertise. And let me show you how that's going to work. Let's go to the internet here. All right, so what I did was I looked at the matches to my great grandfather, Weldon Earl Borland. And this is a reconstructed kit where I've reconstructed 33% of his genome using my Borland Genetics tools. Um, And what I'm going to do, if you look here, there's a column where you can select names of matches. And I'm excluding his descendants, like me and my brother and his nephew. I'm going to do only cousins, and I am going to click his 98 closest cousins. Okay, I already did it because it would take me a while to sit here and do it in front of you. And what I'm going to do next is I'm going to hit submit. And what you get is GedMatch visualization options. For this, to do a cluster diagram, or a heat map, I like to call it, matrices, and we'll go to generations. And by generations, it's going to group them by that number that GedMatch uses to rank your matches by generation. So it's related to centimorgans, but they have their own algorithm. So it takes a minute to calculate if you use all 98 slots, and I highly suggest that you do. Okay, so but I already ran it. And so what we got here is, you see some clusters. And if I go down the diagonal, just like that tool, it's going to do very similar things. And it's going to, you see, it also shows matches that don't cluster in between the clusters. But other than that, it's a very similar tool. Um, And you see there's not a lot of endogamy, I don't think, in this particular ancestor, and that's kind of why I chose him. And I don't know who all these clusters are yet. I mean, I'd have to sit for a few hours and analyze it and look at who these matches are and maybe send out some emails even, uh, or do some quick and dirty or family trees. But, and you see when there is some on the outside, outskirts here, as opposed to just along the diagonal, that is either endogamy or it means that perhaps this group here with the large cluster there, that rectangle, if I scroll to the right, it may just be another branch of this cluster um, with just on a perhaps with a different segment. And if I take a look, maybe I'll know. Yes, I do know who those are. Those are my Burgess ancestors. Now let's see who they seem to be matching up with. Scrolling up, let's look at that nice big rectangle there. Those are my, I don't know who they are. Also with, oh, okay, I see my cousin Valerie. Those are my Walker family. And that makes sense because that's my ancestor, Catherine Walker. Her father was Oliver Walker and her mother was Tabitha Burgess. So they're related. So the reason some of them are probably in both groups is because they are not cousins of my ancestors Oliver Walker and Tabitha Burgess. They're probably descendants, and they descend from both the Walkers and the Burgesses. So that's interesting. Um, all right, but let's take a look at our best example here, which is that big cluster at the beginning. And the biggest cluster isn't always going to be the first one, uh, but in this case it is. So I am considering the cluster to be from, well, from this row all the way down to this row. The next 4 blocks may also be part of that cluster, but I don't know yet. And this part is pretty clear that it is. So if I could figure out which ancestor this cluster belongs to, maybe if I could recognize a couple of the matches. And I do. I recognize my cousin Dwayne, and he's a Smith cousin. And I recognize my cousin Sally, also a Smith cousin. Miriam is also, I know she is a descendant of my Smith family. So these people are all related to me, these cousins, through my ancestor and through Weldon's ancestor, because remember this is his reconstruction that we started off with. So we're starting with someone who was born in 1906. And this is from his, let's see, his mother was Lizzie Miller. His grandmother was Mary Smith, and her father was John Smith, John Perigo Smith. These were all cousins related to me through my great-grandfather's great-grandfather, John Smith. And John Smith was born 1822 in Harrison County, Ohio. So this is somebody that's pretty old that we're going to be able to do a nice reconstruction of using this heat map, the visualization, generations visualization in GedMatch in conjunction with another GedMatch tool, the Lazarus tool. So let me show you how to do that. What I did, and I already got started here because I don't want to waste your time copying and pasting all these numbers, but I called it John Paraguay Smith, and I think I'll put 1822. Segment threshold size, I'm going to use the default settings for this. I don't see any reason not to. He's, of course, a male. We're not going to do any batch processing because this isn't going to be 1,500 centimorgans. I mean, this is somebody from 1820. And we are going to do a reconstructed DNA kit for him, though, in the end of this video. But for Lazarus purposes, let's just do it without the batch processing. Okay, so group 1, I just put Weldon his the DNA kit number for his reconstructed kit. Okay. I've gone ahead of myself a little bit here, but and then all of these kits down here are simply ones I copied and pasted from on this cluster, going down from the 689 to the 071. And looks like I went one too far. 556. Did I do that wrong? Yeah, I'm not convinced 556 is part of this cluster, so I'm just going to get rid of that. All right. So let's run it. Generate the kit. And let me get rid of these since I made a mistake. I'm not going to use those. All right. So not bad, huh? 138 centimorgans. I mean, it's not anywhere near the 1500 threshold. And it looks like there's an error there because it looks like a couple of these are actually in the process of being uploaded. So it may actually be a larger amount of shared DNA after the upload if I ran this again. But anyway, 138 centimorgans, that's pretty good. I mean, if you consider it, that's longer than a lot of our whole chromosomes are. So, all right. So I've got a kit number here for John Paraguay Smith. And let's go to the GedMatch start page and let's take a look at what we got. What I'm going to do is a one-to-one compare to itself so we could see the scope of this reconstruction. 3 segments, a real long portion of chromosome 7. That's really nice that it's continuous like that, that Weldon inherited from his great-grandfather. And then a nice segment that's 58 centimorgans long on chromosome 8, and then a nice one on chromosome 9 too, that's 32.8. So this is really not fragmented very much. These are big, big portions of his genome, which is probably why it has such a nice big cluster like that, because it just at a coincidence, those segments didn't fracture and they didn't recombine in 3 generations for my great-grandfather. So that's kind of cool. Now, let's actually create a kit for John Paraguay Smith. And we're going to do that. We're going to start by going to DNA Painter, and we're going to visualize this. So I've created a brand new profile here, untitled profile, and let's call this my John Smith demo. And I always put what coordinate system I used in the description so I don't forget later. It matters. Build 36 coordinates from GedMatch. Okay, save. Now we're going to paint a new match. And the match we're going to paint is just its match upon itself. It's this box. We're going to take it, paste it, copy and paste. Save the match now. We don't have to worry about excluding segments under 7 centimorgans because we got a nice, we could leave it at that because we've got 3 giant segments. Save match now. I know how I'm connected to this match. Optional, enter the name of your DNA match. Well, I don't know who the name of the match is. It's well, we'll call it Lazarus output, which is the name of my match. Your ancestor's name is John Paraguay Smith, 1822. Is this match on your mother's side or father's? I don't know if I really need to care right now about that for this purposes, but it is Weldon's, no, sorry, it's Weldon's mother's side. I'll choose that color. I'm colorblind anyway, so notes for this group, save match. So if we go down to 7, 8, and 9 now, you see we got the segments painted. And those are the segments that those are some of the segments. Remember, it's only a 33% reconstruction to Weldon to begin with. And on top of that, we only looked at the largest cluster. There may be some other clusters that relate to John Smith as well. There may or there may not be. But this is for demonstration purposes, this is good. So we've got 3 segments there. So what I'm going to do, DNA Painter automatically saves. So I'm going to go to the settings. I don't need to save, but what I do need to do is look at the segment data, and what do you know, it looks just like the output from GedMatch because that's where we copied it from. We're going to save it as a CSV file. And let's open that folder, show in the folder. I've got some interesting things here. Let's see. but I want to open a new folder, a new window rather, and that will be my Borland Genetics data library. Cover myself up here for a second. Okay, and what I'm going to do is I'm going to, where is it? DNA Painter, export John Smith demo. Move it to data library. So now I'm ready to minimize these. And actually, I've got my Borland Genetics fired up already. Manage my DNA resources. What I'm going to do is I'm going to look for that reconstruction because I did the reconstruction using my tools. I'm going to load the resource list that has that reconstruction in it, which is, let's say it's Project Susan. Yeah, Project Susan. Now you'll see at the bottom of the list here, I have a file called Weldon Demo, which is what we're using for this demonstration. And you notice that's 29%, whereas the Weldon up at the top was 33%. And that's just because I needed to use a mono kit for my extract segments tool. And so I eliminated some of the data that was in stereo, favoring the maternal side. Let's go back to the main menu and let's show you how to create John Smith. We're going to go to the extract segments tool, and we're going to, we have them mapped to Weldon demo. Okay, and we use build 36 coordinates in that DNA Painter exercise. And that DNA Painter should be in here. What do we call it? John Smith demo. Okay. So now, which is the target of your reconstruction? We only have one ancestor in the legend, so it is John Paraguay Smith, 1822, which it found from that file. Select, load the DNA resource from the library. So it's actually loading the DNA kit for Weldon. And it's going to extract those three segments. Let's perform some calculations. This could take a few minutes, but not too long. If it was, the entire span of every chromosome, it might take a while, but formatting the output, saving it's already done. It's just saving it to the new kit, to my DNA library. And it didn't ask me what to save it as because it's just going to save it as whatever was in the legend, which is John Paraguay Smith. Male. Okay, return to the main menu. Let's take a look. Let's do a visualization in my toolkit in the segment view export section. And let's take a look at, there it is, John Paraguay Smith, 1822. It added it to the bottom. Select, and let's render the visualization and see what segments were reconstructed and make sure it's the correct ones or extracted. There they are. And it should look just like these three segments. And go back to my tool. A lot of windows open right now. And it created exactly those three segments. So what we could then do if we want is go back to GedMatch. Let's go to the home page. Log in. Generic upload fast. Name of donor, John Smith. And male. I have no idea what his haplogroups are. I have permission. Artificial kit. Choose my file. It should go to my data library. It doesn't. Okay. And we'll go to, it created that John Smith John Perigo Smith zip file. The design's ready to go to GedMatch. And we're going to upload, aligning. All right, now let's put it to the test. We got a Z number, right? Okay, so let's go back to GedMatch. I'm going to go a 1-to-1 compare, see if this worked. Got my Z, and now go down to my Lazarus kits there. I got John, this is the one we just created, right? John Perigo Smith 1822. Let's compare them. Do a 1-to-1. And I'm just going to do position, submit. Not bad. They have 4 matching segments between the Lazarus version and my Borland Genetics version. And it looks like there's just a slight gap between 50.3 megabase pairs and 50.4 megabase pairs on chromosome 7, but then chromosome 8 and 9 have those nice big matches. But I mean, it's a different algorithm that the Lazarus tool is compared to my tools. If we waited, then my zkit would process overnight, and we'll see the matches. I'm going to do that because I'm curious what's going to happen. Whether I get a lot more than the matches that showed up in those clusters or not, and I probably will because I only chose the top 100 of Weldon's matches, and out of that only used a small fraction of them for that to define that cluster, whereas he's probably got a lot of smaller matches along that cluster as well. And maybe they'll have some structure, and maybe I can repeat the process and perhaps even make a Lazarus kit that distinguishes between the ancestors of, from the descendants and cousins of John Perigo Smith's father, Daniel Smith, and mother, Elizabeth Perigo. And that would be kind of cool to take it back another generation. And I'll see if I could do that, but I have to wait till tomorrow. So until then, it looks like it was a successful reconstruction, and we used the coolest, latest technology similar to that being used in the genetic affairs tool that everybody's been talking about. See you next time.